So I'm going to talk to you about one of my favourite subjects, which is systemic lupus erythematosus. And I'm going to do this from the point of view of trying to illustrate to you how investigators have used some genetic models to find out what might underlie the pathogenesis. So I would say that invariably, to tell you something about this, I also have to teach you something about immunology, okay? Because otherwise you won't be able to understand the relevance of some of the genetic factors that have been found. Okay, so what we're going to cover over the next hour or so, firstly I'm going to tell you how various definitions that relate to genetics and rare diseases apply to lupus. I'm then going to give you a typical clinical case, so this is based on somebody that I've looked after in the kidney clinic who has lupus, to give you an idea of the impact that this disease can have. So it can be a pretty devastating disease. So I'm then going to talk to you about how we can, things that we can surmise from the clinical features about the etiology, then talk about pathogenic mechanisms and particularly focus on how mouse models have allowed us to define some aspects of the pathogenesis. Then talk about abnormal B-cell activation, and then finally how we've used some of the information from mouse models to define new therapeutic options for patients with lupus. So you mentioned that previous sessions perhaps hadn't been long enough. I don't think you'll say that about this one, because it is a subject of which I'm extremely enthusiastic about, which I'm sure you're very glad about. Okay, so in terms of definitions, so you're here as rare disease MPhil students. And you will have heard already that we define a rare disease as a disease that affects less than 1 in 2,000 of the population. And so formally there are at least 6,000 diseases that have been defined as rare diseases. But you can see that that would change depending on what your baseline population is, because there are some diseases that are more common in some populations. And overall, although these diseases individually might be relatively rare, they affect a lot of people in the UK, so more than 3 million. So lupus is a polygenic autoimmune disease, and you've heard a little bit about one of these already. You've heard about inflammatory bowel disease. And what we mean by that is that unlike monogenic diseases, for someone to get lupus, they have to have abnormalities or genetic variants in more than one gene that together contribute to the development of disease. And so these individual variants are often, if you looked in the general population, they are often relatively common, but it's only if you line them up, and in isolation they don't necessarily cause disease. So it's only if you line them up in a particular way that they will cause disease in an individual. So for lupus, it's rare, so it affects about 3 in 10,000 people in a Caucasian population. Actually, for a Southeast Asian population, it's more common than that, and that's driven by genetics. In terms of the genes or the loci that have been implicated, there are more than 50 susceptibility loci that have been implicated. So you can see that if I was to describe the studies that have been done for each of these, we'd be here a very long time. So I've just taken a sample of each of these. Okay, so why do people find this disease interesting? Why do I find it interesting as a clinician? So to convey this to you, I'll give you a clinical case. So this is a 24-year-old lady who presented with a rash. And this rash had principally been across her face, but also she'd noticed some rash on the back of her hands as well. So in, you could say, sun-exposed areas. She'd had some intermittent chest pain, which she described as sharp. And in medicine, we know the significance of that is that sharp chest pain that's worse when you breathe in is called pleuritic chest pain. And it usually means there's some sort of inflammation in the pleural membranes, so pleurisy. And she'd also just generally not been feeling well for about 3 months. So she'd had lethargy and also joint pains, particularly in the small joints of her hands. So as is usual, we go on to take a history from patients when they come to us with a problem. And that includes asking about their past medical history. And actually, this lady was relatively young and had previously been completely fit, so had not had problems. The only medicine she was taking was the oral contraceptive pill. and she actually had no family history of disease. So you will have learnt already that it's important to take a family history in patients who present with diseases, so particularly ones that you think might have a genetic etiology. So this is the rash that she had. And this sort of rash has been dubbed as a butterfly rash, so you can see why it sort of has the shape of a butterfly. So we went on to do a number of blood tests, the sort of simple blood tests that you do. One of them was to look at whether she was anemic, and in fact she was. Her white blood cell count was at the top end of normal, and her platelets, which are responsible for helping the blood clot, were on the lower side. But what was more worrying was when we looked at her kidney function tests, and to do this we look at something called creatinine, which is a waste product that's removed by the kidneys. And so therefore, if your kidney's not working so well, the level of creatinine goes up. And when we looked at the level of that, that was actually higher than it should be for someone of that age, suggesting that her kidney function was impaired. And to go along with that, when we dipped the urine, we could find both blood and protein in it. And those things are, although that seems like nothing too much, that's actually highly significant in a young female and suggests there might be inflammation. So given that history and those findings, we suspected that there may be an autoimmune disease going on. So we went on to do a number of tests. So that included an autoantibody screen. And this is the first test that's done. So this is a test for anti-nuclear antibodies using HEP2 cells. So these are a human epithelial cell line. So what you do is you grow up your cells so they're confluent. You then put the patient's serum onto the cells in a way such that the cells are permeabilized. Then if the patient's serum contains antibodies that can recognize nuclei, they will bind to the nuclei of the cells that are on your slide. And then you come in with a secondary antibody that recognizes human IgG. And all that will be, there will be whatever's bound. So you can see very obviously that the antibodies that this lady had were antibodies that bound to the nuclei of the cells. So these are anti-nuclear antibodies. And this test is sort of highly sensitive for lupus. So about 95–99% of patients with lupus will have anti-nuclear antibodies. But interestingly, it's not that specific, because actually when you look at people who are old, so patients over the age of 70, about 10% of them will have ANAs. But in this group, it's pretty sensitive and specific. And if we looked further at the autoantibodies, we could see that she had some antibodies that not just recognised generally nuclei, but specific nuclear components, so double-stranded DNA. As well as this, she had high levels of IgG, which is a circulating antibody, and low levels of complement, and I'll explain to you the significance of those things. So to understand what I've told you there, I just need to briefly tell you something about antibodies, okay, because otherwise you won't be able to follow what I'm subsequently going to tell you. So you probably will have all done some immunology at some point in the past, and it may be more or less hazy in your mind. But antibodies are made by terminally differentiated B cells. Okay? So B lymphocytes, normally they express an antibody on their surface, and this antibody can bind to very specific substrates called antigens and activate that B cell. And if they do that, it will differentiate to become a plasma cell. And plasma cells are essentially just antibody factories. They just keep producing antibody. And these antibodies have got two domains, so they have a variable domain at the end, at both ends of the Y, and those bind to specific things. So if I took your blood and had a look at what things your antibodies would bind to, your antibodies could bind to about 10 to 11 different specificities. Okay, so you have an amazing capacity to recognise things with antibody. And it also has a constant domain which doesn't vary between antibodies, and both of those play an important role in the function of antibody. So I'll give you an example. We make antibody because it's part of the immune system, it's part of an immune response to pathogens, and antibodies can directly neutralise pathogens, so that they can use these variable domains to bind to pathogen or toxins to stop them from causing trouble, invading cells, etc. But if that's all the antibody did, they wouldn't be that powerful. What makes them more powerful is the fact that they can recruit other parts of the immune system. So the first thing they can do is activate complement, which is part of the innate immune system, and they do this via the classical pathway. And complement is basically a series of proteins that's made in the liver that can be sequentially activated, resulting in the formation of a pore, which if it's punched in a bacteria, you can see that's good. If you activate this against your own cell, very bad, because it will punch a hole in the cell. And you can see the different things that are in that pathway, so C4 and C3. And I mentioned to you, in patients with lupus, if we look in their serum, they have low C3 and C4 because this pathway is being driven. And then the other important things that antibodies can do is that they can bind to surface receptors, and these are called Fc receptors, and that makes sense because they're binding to the Fc bit of IgG. And in the case of IgG, they're called gamma receptors because the gamma chain is present in IgG. And these are really important because almost every cell in the immune system has Fc receptors. So by virtue of binding an antibody to something, you can activate natural killer cells, neutrophils, macrophages, DCs, B cells. So this is a really powerful way of switching on the rest of the immune system. So you can conclude from that antibodies are not just effective because they can neutralize things by their variable domain binding, but because they can switch on complement and activate other cells via Fc receptors, they can really get an immune response and inflammation going. So you can see that if you have an autoantibody that recognizes one of your own cells, and you can engage all of those aspects of the immune system, this is bad news and can cause a lot of inflammation and damage pretty quickly. And if we look, so this was a study that was done looking at the different sorts of autoantibodies that patients with lupus have. And as well as we talked about the DNA antibodies, and that's the DNA antibodies is something that we look for routinely. We don't look for all these things routinely. But you know, other examples are for example, this one. There are antibodies that recognize red blood cells. So if you recognize a red blood cell, you punch a hole in the membrane, the blood cell gets destroyed. Patients can have very severe anemia that's even life-threatening. Similarly, platelets. So our lady had slightly low platelet levels, and actually she ended up having an autoantibody that could recognise her platelets. But what I want you to there's a whole range of things. I don't want you to remember them individually, but broadly speaking, we can say that many of these are nuclear components. Okay, so there are both DNA and RNA antigens there, as well as other nuclear components. And that's, I suppose, one of the very unique things about lupus. So going back to our case, because she had kidney dysfunction, so as kidney doctors, there's nothing we like better than doing a kidney biopsy because it gives you the diagnosis. So we went on to biopsy this lady, and so on the left-hand side there I've included a normal kidney biopsy. So that shows a glomerulus, which is the filtering unit. And it has, you can see these little loops. Perhaps it'd be easier if I put my thing on there. You can see these little capillary loops, and they should be very thin because you've got to squeeze blood out of them. Okay, but you can see in this one here, which is the lupus biopsy, these capillary loops are no longer thin. Look at this one. They're really thick and fat, and that's called wire looping. And that happens because what's deposited there, making things thick, are antibodies. So if we stain specifically for IgG, we can see that the glomeruli are lined with this IgG. And we can also see evidence of complement activation. So if we stain for C1q or C3, we can also see those. So the diagnosis here wasn't that difficult, and we were able to say that she had kidney involvement, which actually has significance for her prognosis. And the way that we treat these people, or certainly treated them up until perhaps about 5 years ago, was very unsatisfactory. Okay, so I've said the average age of development of this is 29, 30. And these young, relatively young people, we're giving things like cyclophosphamide, which is something which prevents new DNA synthesis or wipes out cells that are proliferating. And things like cyclophosphamide carry with them a long-term risk of cancer, so particularly cancers of the immune system. So it's not good having to give young people these sorts of toxic, non-specific drugs, which is what we've had to do. The other mainstay of treatment is corticosteroids. And corticosteroids, basically, if you give them in high dose, make people put on weight. They make them diabetic, they make them hypertensive, and they make them osteoporotic. And all those things mean that it sort of had got to a point with patients with lupus and other autoimmune diseases where the effects of the treatment, the side effects of the treatment, were more likely to kill you than the disease itself. So I'm going to talk to you about some of the things we've learned about the genetics, but those have actually informed our treatment strategies. Okay, so these are the diagnostic criteria for lupus. You can see Our Lady had some of these. This was the most recent classification. But the nub of it, so learning point 2, is that lupus is a multi-system disease. So it can present, with just a rash and a bit of joint pain through to life-threatening kidney failure. So in that way, I guess it's a very interesting disease that can present to many specialists. And underlying it, these patients have IgG autoantibodies that deposit in tissues and activate the immune system by the ways I've told you, complement Fc receptors. And I would say, so because it's a multi-system autoimmune disease, it's one of those diseases that sometimes it takes a long time for people to diagnose because, some of the symptoms are, I just don't feel very well, I'm a bit achy, I'm a bit tired. And in younger people and females, so they're often deemed a bit mad before eventually someone does some investigation and finds out what's wrong. Okay, so the etiology. I'm going to first of all give you a couple of slides about the background, the historical background, and I would say that just reflects my interest. When I look at a disease, I always like to know how people have approached it historically. So lupus is actually named after the Latin term for the wolf, Canis lupus, and that's said to be in some of the early literature because the rash that some of these individuals got, so on the left-hand side, in an era before you were able to treat it, was so severe that it looked as if a dog or a wolf had attacked the patient. And it was first appreciated that what we now know as SLE was different from other forms of severe facial rash by Cazenave in 1851. And he distinguished the difference between two, well, certainly this on the right was a relatively common seen thing. So lupus vulgaris, which is cutaneous TB, but you can see a similar sort of terrible facial involvement. And what he called lupus erythematosus. And then Kaposi later on appreciated that this wasn't just about rashes, that patients had more than that. They had fever and joint pain. And then finally, a couple of clinicians realised that this really was a multi-system disease, so particularly that there could be kidney involvement, which was significant. So what we now know is that this is a disease, as I said, a mean age of about 30, but this is a predominantly female disease. So of all the autoimmune diseases, this one has the strongest female association, so 9-to-1 female to male. We also know, as I said, that it's more common in both Southeast Asians and Afro-Caribbeans, but not just more common, but more severe. So they're more likely to have renal involvement and be difficult to treat. We also know that certain drugs, so certain environmental factors, can induce a lupus-like disease, and that's been interesting to investigate, so particularly a drug called hydralazine. And then finally, although in our patient there was no family history, it has been shown that there is a familial association in that patients who have lupus are more likely to have a first-degree relative with lupus, or significantly a first-degree relative with some other autoimmune disease. So, we're interested in this course in genetics and the role of genetic variation. So why did we start off, why did investigators start off down the route that there was a genetic underpinning to this? Because you can see, as patients like the one I presented, they might present with no family history whatsoever. Well, we've started on this because, first of all, we know from larger epidemiological studies that siblings have a 15 times increase of developing lupus. Okay, so that's the sibling risk. And we also know if we look at dizygotic twins and compare that to monozygotic twins that are identical, the rate of concordance, so the likelihood of getting disease, is 10 times higher. So 30, if you have a twin with lupus and you're a dizygotic twin, 3% will get disease. If you're a monozygotic twin, 30% will get disease. And then the other thing, as we know, there's significant racial variation, and that holds independent of environmental background. So if you take someone of Southeast origin and move them to a population that's predominantly Caucasian with a different environment, the same risk holds. So that suggests that there is strong genetic etiology to it. And we now know, to summarise, that, as I said, it's polygenic, more than 50 different variants. They're common in the general population, but individually don't cause disease. But together, if you line them up, they can cause disease. But we also know that it's a disease that has environmental inputs that might make the appropriate milieu for those genetic susceptibility factors to actually trigger disease. And that includes hormonal factors, and I don't have time to tell you all about this. There's lots of research on the effect, for example, of estrogen on immune cells and how that works. But also other environmental factors, so I mentioned drugs like hydralazine, but also infections, particularly some viral infections have been shown to trigger disease, and sunlight can trigger disease. So that, you know, I described to you that lady had a rash in the areas of sun exposure. And again, we now understand why that is, okay, and I'll perhaps allude to that a little bit later. Okay, so how can we identify the genes that are involved, broadly speaking? So a genome-wide association study, which is what we can do now. When people started investigating this, that wasn't available, but a genome-wide association study where basically you take a group of patients with disease, a group of controls that are of the same racial origin, and you have the capacity to look for the presence of literally hundreds of thousands of different SNPs, so single nucleotide variants, and you're able to say, are there some that are more common in the cases and more common in less common in the controls? So to date, I actually was going to tell you about some of the GWAS stuff, then I cut it out. I already had too much to tell you about, but there have actually been now more than 10 GWASs, so, and this has really happened in the last, 5, 6, 7 years. There have been more than 10 GWASs that have looked at lupus populations on various different genetic backgrounds. And perhaps I'll just summarise and say that unsurprisingly, one of the key variants that's come up are variants in MHC, so class 2. Which are involved in antigen presentation, and I'll tell you a little bit about that, why that might be important later. But when people started this, GWAS technology wasn't available. Okay, so all you had was a collection of patients with lupus and a collection of controls, and I'm afraid to say, sadly speaking, there are also often a very small collection of patients with lupus and a very small collection of controls. And the difficulty with doing those sorts of studies is you're very likely to get things wrong, and that if you go back through the literature, things that were said to be associated now clearly aren't. But I'll tell you about some of the candidate gene studies that were helpful. So as I've said, we've now had GWASs, and now we're able to interrogate someone's genetic variability in even more detail by doing whole exome or whole genome sequencing. But that hasn't greatly been applied to this. But I think in future it probably will be, because it's now becoming, rather than just looking for specific SNPs as the GWAS does, although it's hundreds of thousands of specific SNPs, actually being able to individually look at someone's variability will be cheap enough to do. And you've heard about the 100,000 Genomes project, and you can imagine that being rolled out eventually when it's cheap enough. Now, what I would say that perhaps isn't obvious, but maybe you've thought about this, is that these are, when we say, I wonder what genetic variant might be involved in a disease, these are ways in which we can directly look at the variation in the genes. Okay, these things won't cover is epigenetic variation, and you had a lecture about epigenetics, and that's another field. But we can also get some idea about genetic variation if you think about it indirectly. And actually, many times in medicine, we have started here with protein biomarkers, because if you think about it, right, what we're trying to look at is genes that are transcribed and then translated to become proteins. So you can actually assay someone's proteins and say what's different, and that can give you an idea about genetic variation. And you might argue that unless a genetic variant has an impact on the level or the type of protein, that it may not matter, and that's probably true in lots of cases. So I think studies that look at proteins can also be helpful. And then also, you can indirectly look at genetic variation by looking at the mRNA species. And people have done that now quite extensively, because you can again do it on a platform where you can look at hundreds of thousands of different transcripts. And those, you know, I've got to say transcriptomics, and you can do transcriptomics on peripheral blood, and people have done that in lupus. And you can do it on biopsies as well, and again, people have done that. And that, again, I don't have time to tell you about those studies, but really those have also, using the transcriptome to tell you something about genetic variation, has been useful in lupus and identified the type 1 interferon signature. So there's a paper from Bonchereau and colleagues in the New England Journal a long while ago now. But so I think when you're thinking about how you can assay a person with disease in terms of their genetic variation, don't just think about directly looking at genes, but think about the fact that both the mRNA and the protein can also give you an insight into genetic variation. So the other models that we use, and this is what I really want to focus on in this talk. And I would say in lupus, I would argue that probably mouse models have identified more genetic variants that contribute to disease than other studies, than human studies. So I've distinguished here two things, and you might say, well, what's the difference between those two? So one is mouse models, and the other is knockout mice. And what I mean by that is there are actually some mice that spontaneously develop a lupus-like disease. So you could see them a bit like people that in a polygenic way spontaneously develop a lupus-like disease. But then there are also some mice that if you knock out a specific gene and you stress the system a bit, you can make them develop lupus. So those are two slightly different ways of looking at it, and I'll describe that in more detail. Okay, so key fact 3, you're all still with me. So the pathogenesis of lupus, there are both genetic and environmental factors. And the way that we can look at this are human genetic studies, and that might involve looking at genes, mRNA or protein, but also mouse models. And that's identified, like I say, it's probably even up to 100 variants now. Okay, so pathogenic mechanisms. How have we found out which genetic variants contribute to pathogenic mechanisms? So if I was to summarize the pathogenesis of lupus, and this is obviously something that I've thought about a lot, I would break it down into 4 basic stages for simplicity. Okay, so the first one is that there must be a breakdown in immune tolerance to self. So the idea was proposed 50, 60 years ago, that our immune system has developed such that it doesn't recognize our own antigens, our own proteins. And clearly in this disease, these individuals are not tolerant, which is what that term means, not recognizing your own proteins. So this leads to the generation of autoantibodies, which we can see. These autoantibodies are deposited as immune complexes, which just means a combination of antibody and antigen. And we know in lupus that first of all there's a failure to clear these immune complexes, but also the immune response to those immune complexes is excessively exuberant. Now, if we're thinking about the breakdown in self-tolerance point, the first part of this, our clinical observation actually tells us a really critical thing. So these autoantibodies, I told you that they're IgG autoantibodies, they're a particular isotype. And that actually has a significance. It tells us something already about what must be contributing to the breakdown in self-tolerance. And these antibodies are not only IgG, so what's called class-switched, they're also what's called hypermutated. So if we looked in their variable region genes, we would find many mutations introduced into them compared to baseline. And those two facts tell us, we can surmise from those two facts that to produce these autoantibodies, there must have been not just an autoreactive B cell, but an autoreactive T cell. And, you know, as I said, the dogma would be that you shouldn't have either of those, right? You should have got rid of those during lymphocyte development. And I'll tell you why we can surmise that, because you're probably thinking that's not obvious to me why we can surmise that. So this is, so this cartoon that I've done here, this summarises what happens when you activate a B cell. Okay, so what I'll point out to you is that, so I'm activating the B cell with the antigen, which is that black blob. And you can see it's binding to the surface antibody. Now, the antibody that a naive B cell expresses is an IgM antibody. So that is the chain, the heavy chain that it has is a mu heavy chain, so it's an IgM antibody. Once it's bound to that antigen and becomes activated, it will then, so B cells generally, they circulate around the body, but they live in lymph nodes and spleen, and that's where they get activated. So this B cell in a lymph node will then go off, it's got its antigen, internalise it, it will chop it up, and it will load it onto a protein called MHC. And it will hold it on its surface and say, I'm looking for help. I'm looking for help in response to this protein that I've chopped up. And the help that it wants is help from a T cell. So it moves from the follicle out to the edge where it will find, because T cells live in this zone and B cells live in that zone, it'll go say, hello, is there a T cell out there that wants my help? And the T cell will come and it will recognise that antigen in the context of MHC. And then if it gets that signal, if we focus on pathway 2, the B cell will go back into the follicle and it will undergo what's called a germinal centre reaction. And for this, it needs T cell help, so specific T cells that are called T follicular helper cells. Now that makes sense because they're T cells, but they live in the B cell follicle and their job is to help T cells, so they're B cells, so they're called T follicular helper cells. So if a B cell has got its antigens, presenting it, it's getting help from T cells, two things happen to it. One, it class switches. It changes from expressing an IgM to expressing an IgG. So do you see why we can conclude, if we see IgG autoantibodies, that B cell, that autoreactive B cell, must have had help from an autoreactive T cell that was recognising the same antigen. So it tells us that in that person that's developed lupus, they have an element of faulty tolerance, yeah, because they've allowed some autoreactive cells to persist, which shouldn't happen, although we know does. Or either that, the ways of suppressing those autoreactive cells, because actually we all have, we know now that central tolerance is leaky, we all have autoreactive cells, but we have many ways of keeping them quiet. And clearly in these individuals, the ways of keeping autoreactive B cells and T cells quiet have gone wrong, or else we would not see the IgG. So the other thing that happens, the whole purpose of a germinal centre reaction, right, is to take that antibody that the B cell's making and to make it better. And the way the B cell does that is by introducing mutations into the variable gene so that it has a higher affinity for antigen. So we can identify in those autoantibodies in lupus patients mutations. So we know that it's been through this process, so T cells have been involved. Okay, so with that in mind, we can assume that at the breakdown of tolerance, we unfortunately have both an autoreactive B cell and an autoreactive T cell, and that T cell will have had antigen presented to it, so it will have been activated. And antigen presentation is a very specific process. And I just touch on this because one of the genetic mutations I'm going to talk about involves this process of antigen presentation. So an antigen presenting cell, for example a dendritic cell, would take up antigen, internalise it, like a B cell's an antigen presenting cell too, chop it up and then load it up onto class 2. But at the point where it internalises into endosomal compartments, there are also receptors there that can recognise things about that antigen. So, for example, there are receptors that can recognise whether there's something on that antigen that indicates it's from a pathogen, because you can see, you know, then this antigen presenting cell needs to tell a T cell, what I'm holding you here is something bad because I've detected it's from a pathogen. So the antigen presenting cell will then not only hold up the antigen, but it will also hold up a co-stimulatory molecule that will really drive that T cell. So this process of antigen processing and presentation and the recognition of things that are foreign signals during this pathway is really important. And if it goes wrong, so that the antigen presenting cell gets confused and says the thing that I've internalized I think is dangerous and actually it's a self-antigen, you can see it will give the wrong signal to the T cell. And that's one of the things that happens. Okay, so it means that the T cell then gives help to the B cell in the way that I described in the germinal center response. That B cell will proliferate, it will form a long-lived plasma cell and produce lots of antibody. And this antibody, if it binds to antigen, will form immune complexes, and this is what happens in lupus. But we all actually, so people have done studies, if I sampled your blood, I would be able to find some immune complexes, but actually we normally have ways of disposing of them. So we know that in patients with lupus, again, there can be a problem with the disposal of immune complexes so that they hang around and deposit in kidneys. And then the other issue is that when those immune complexes, which should have been cleared but they aren't, if they get deposited in the tissue, again, we have ways of dampening inflammation so that not every insult causes horrible inflammation. But in patients with lupus, they will have a genetic variant that somehow means when immune complexes get deposited that they respond to that too much in an exuberant way. So loss of tolerance, generation of autoantibodies, a failure to remove them so they get deposited, and then excessive inflammation to immune complexes are the 4 key steps in pathogenesis. So how do we find out why all of those things happen? So why is there a loss of self-tolerance? Why is there deposition? Why is there excess inflammation? So if I take you back to these key things, this is really what's been used to answer those questions. And I'm going to start with some of the mouse models. So the mouse models of lupus, there are 3 inbred strains of mice that have been used as models that spontaneously get lupus, and these are these ones, the MRL mice, the BXSB and the NZBWF1, so the first generation of the cross between those two. Now, you might say, well, how did these things come to light? Now, in my PhD, I was looking at a mutation in a particular receptor that predisposes you to lupus, and I'm going to tell you a bit about that. But as part of that, I actually did quite a lot of reading as to why some mice that we looked at had this mutation. And this got me sort of trying to understand a bit about where inbred strains of mice come from. So it turns out that at the turn of the 20th century, there were a group of individuals initially in China and Japan who were termed as mouse fanciers. And they collected mice as pets because either they looked quite pretty, they had a coat that was a nice colour, or they did funny things. So for example, there was a mouse called the waltzing mouse that just sort of went around in circles. And they then bred these pretty coloured mice together, and some of them did funny things and some of them not. And it's actually from those mouse fanciers that our inbred strains of mice come from. And I was heartened to see that the mouse fancying business still goes on. So this is a picture taken from one of their conferences where this mouse here was deemed as best mouse that can poop, as you can see there. Okay, so this is I mean, I never imagined that eventually all this reading would come to a paper, but it did actually. So a long time after my PhD, we finally got all of this together, and this is a summary of a family tree of inbred strains of mice. So I actually got this I derived it from a Nature paper, so that was published, I think, in about 2003 by Beck et al. And they had an enormous poster that literally documented every single inbred strain. So that would be a conversation in the pub for you. But what you can see is that, so there's our Japanese mouse fanciers, and then there were some English mouse fanciers. But you can see that there's this character Lathrop here. So this was a lady called Abby Lathrop, who was a mouse fancier who lived in Massachusetts, and she collected mice. And actually, I suppose, well, as women then, you couldn't formally get involved in science, or it wasn't really the done thing for you to do PhDs or. But she actually was interested in experimentation and collaborated with 2 people who worked at Harvard. Granby and Little and Castle, and gave some of her inbred strains that she had bred in her garden or whatever for use for mouse models. And actually she contributed to some papers that defined the effect of hormones or the endocrine system on some cancers. So this is where the inbred strains come from. And you'll see the ones that I've mentioned, well, perhaps you won't see on this, but the MRL-LPR mice, yes, that's one of the models. So you can see we can trace those right back up to there. The BXSB mice, so there they are, and you can follow those through. And then the NZBWF1, so they were actually got from some European mouse fanciers and then developed in Mill Hill. So this is where all our inbred strains come from, and sort of actually just randomly by inbreeding, it means that recessive mutations that can come together in a particular combination that lead to disease. And I'm going to focus on one of these that it turns out reveals to us the importance of antigen presentation and how things can go wrong in antigen presentation. So that's this one, which is the BXSB mouse. So what experimenters observed was that if you crossed a black 6 mouse, which is just a standard controlled strain, if you cross a female black 6 with a male SBLE, which is another one of the inbred things, you can look at the family tree, the resulting offspring, which were termed BXSB, got autoimmune disease. So they developed anti-nuclear antibodies and had other features of lupus. But they found that if you did the cross the other way around, so used a black 6 male and to a BXSB female, those mice didn't get accelerated disease. So this observation led to the concept that there must be something on the Y chromosome that was an autoimmune accelerator. So what investigators did, so this was done by Sylvia Boland's group, was that they said, okay, well at this stage, so this was back in 2006, how can we identify what's different about the Y chromosome in these mice? Because that's probably going to tell us the key, what's the genetic contribution to disease in these animals. So what they did was use fluorescence in situ hybridization, where you use probes that recognise specific sequences that are found on bits of chromosomes, and they're fluorescently labelled so you can see them. And hopefully you can appreciate, so here's a black 6 mouse on the right, and here's our BXSB derived mouse there on the left. And what you can see is that the probes are lighting up on the end of the X chromosome, yeah, can you see in red and green? And in the BXSB mouse, those same probes are binding to something on the Y chromosome. And they were able to look at that in a bit more detail. So I've blown up the chromosomes there so you can show them in more detail. So whereas if you look at the black 6Y, it doesn't light up because it doesn't have those sequences. In the BXSB, it does. So it has a bit of the X chromosome bolted onto the end of the Y chromosome. And apparently that's not uncommon in humans too, to have some translocation between X and Y. And they use these probes, so there are various different fish probes that you can use that allow you to walk along the X chromosome and say which bits of it are there and which are not. So you can see the probe there, the 12517, which I put an orange box around. And then this shows you the DNA and what genes are known to be in that part of the DNA. And you can see these were the two probes that lit up. And one of the genes that we can see here is something called TLR7, or toll-like receptor 7. And the investigators that were looking at this were very interested in this because toll-like receptors, there's been a great deal of interest in innate immunity, and in fact, the discovery of the toll-like receptors earned a Nobel Prize for a couple of people a few years back. So we now know that there are actually 11 of them in mammals, and they're what's called pathogen recognition receptors. So they recognise repeating molecular patterns that are found on the surface of pathogens, or that are more pathogen specific. And TLR7, you can see, actually recognises what you hope is viral single-stranded RNA. So if we trigger the bell to lupus patients have autoantibodies that recognise RNA. Now here's one possible way in which things can get confused. Okay, and so they went on to say, okay, we can see that there's a bit of that chromosome on there that contains TLR7. is there more TLR7 message in these mice? And by using RT-PCR, they could show, yes, there was more TLR7 message. And then they used Western blotting to confirm that there was more TLR7 protein as well in these mice. And then they were able to show that if you treated these mice with imiquimod, which is an agonist of TLR7, you could make the type of autoantibody that they produced, the ANA that they produced, more nucleolar. So that is recognizing things that were more likely to contain RNA. So you could actually change the type of anti-nuclear antibody. And this group has now published a whole range of papers showing how in dendritic cells specifically, if you take away everything else and make a mouse that only has too many copies of TLR7 in their dendritic cells, that is sufficient to cause a lupus-like disease. So having a system where you're just expressing so much of one of these pattern or pathogen recognition receptors that the danger is it can appropriately recognize self-nucleic acids is sufficient to break tolerance. Okay, so that's example 1. So example 2, I'm going to show you a couple of examples of knockouts. So this is now not mice that were bred and spontaneously get disease, but this is mice where a specific gene has been knocked out based on other observations, or on a hypothesis. So I use this to just remind you that knockout mouse models are an incredibly powerful tool. So I think someone from the Sanger came to talk to you about mouse models, and they may have mentioned this. I hope they will have mentioned this. So this is an absolutely extraordinary program that's going on. Is a European NIH sort of consortium, and what they're aiming to do is to make knockout mice for every single gene in the mouse genome. And when they make a mouse, they run it through a screening panel that includes, they infect it with some things, influenza and salmonella, but they also screen every one of these mice for the development of anti-nuclear antibodies to see if they can find things that increase susceptibility to autoimmunity. So far, they have a little website, and this is centrally funded, and they've actually made 4,000 knockouts so far, and they're aiming, they hope that they will have knocked out all 16,000 genes by 2021. So the power of these is that you can really say in vivo, in an animal model where you've got multiple, complication of multiple things, does my gene play a role? And you'll notice that one of the things that it allows you to do, so it actually has a rare disease models tab that you can go to, and you can type in whatever rare disease you're interested in, and it will give you what mouse models might be relevant to that if you're interested in investigating things further. So one of the mouse models, I'm going to talk to you about 2 of them, so DNase 1 knockouts and Fc gamma R2b knockouts. And I'll show you how knockout mice, which showed a phenotype in developing lupus, led to studies in humans and the identification of mutations in humans. And you need to go further than that, because we all have mutations or variations everywhere. You need to show that that mutation is of functional significance. And so these are 2 examples where, in part at least, for the first one that was done. So the DNase 1 knockout mice, if you let them age, they spontaneously develop anti-nuclear antibodies and actually anti-DNA antibodies and develop an autoimmune disease. And this led a Japanese group, and this was published in Nature Genetics in 2001, to do a case control candidate gene study. So they said, well, we've got this interesting knockout mouse that has this phenotype. I wonder if in our cohort of lupus patients, there are any mutations in this gene. Now you can see, remember, just going back to one of the problems with olden days genetics, candidate gene studies where you've got 20 patients with disease. If you compare that to GWAS studies where you typically have thousands or even tens of thousands of patients, you can see why this can be prone to error. But anyway, they were lucky in that they only looked at 20 patients, but they identified that 2 of these patients had nonsense mutations in exon 2 of the DNase 1 gene. And they went on to do some functional studies showing that the effect of this mutation was to reduce DNase activity. And we know that DNase is an enzyme that is responsible for breaking down DNA that's released. So the idea is hopefully your immune system never gets to see DNA because this enzyme is chopping it away, breaking it down so that it never is an antigen. But if your enzyme doesn't work, it means that DNA can persist, so you actually have abnormal antigen exposure, whereas normally it'd be got rid of. So therefore, if you do happen to have an autoreactive B cell or T cell that can recognise this, this would be sufficient to trigger an immune response. And the way in which they looked at this further was to take their 2 patients with their DNase 1 mutation and then look at other patients and some healthy controls. And they looked at the level of double-stranded DNA autoantibodies these patients had. And you can see that those with the mutation had 10-fold the antibody, double-stranded DNA antibody titer, so you can see there. Whereas if you compared their antibody response to something else, so for example they'd all been tetanus vaccinated, it was not different to the first column. So showing that this specifically, if you don't break down your DNA, if you've got a polymorphism or a mutation in this, then you're more likely to have DNA that accumulates that can be recognised by the immune system. So if you do happen to have autoreactive cells, you'll develop antibodies. Okay, and so although these, I guess, I would say my point would be that, these people had nonsense mutations, which meant they had 0 DNase activity. But there are likely to be polymorphisms that mean individuals have different levels of DNase activity, and that might be sufficient to allow exposure of self-antigen. Okay, so what I've given you there is 2 examples of a mouse model. So one which is a polygenic spontaneous lupus model, and the other is a knockout mouse model that have been allowed us to determine some of the underlying contributors. So I'm now going to go on to part 2 of the lecture. The second thing I'm going to talk about is what's fair to say is my favourite protein, okay? So we'll put that up there as a caveat right from the start. So Fc gamma R2b, so you can see Fc gamma R, so it's a receptor for the Fc portion of IgG. Okay, so immediately you can see why it might fit into lupus. So I showed you that Fc receptors are expressed on a whole range of cells earlier, right? I showed you the diagram where they're expressed on NK cells, neutrophils, dendritic cells, macrophages, but also on B cells. And actually, B cells only express this Fc receptor. And the critical thing you need to know about it, that unlike all the other Fc receptors, this receptor is an inhibitory receptor. So what it can do is when a B cell, so you can see my little B cell here, and I've drawn this is the BCR, right? This is his surface antibody. So the antigen will bind to there, and then B cells, like T cells, have a whole other lot of co-activating receptors. So for example, if there's complement around, that will bind to complement receptor 2, and that will make this cell easier to activate. So they modulate the threshold of activation. But B cells also have inhibitory receptors, and what they can do is recruit phosphatases, which switch off cellular signaling. Yeah. So all cell activation basically requires kinases which phosphorylate things, more or less. So what these inhibitory receptors do is recruit phosphatases that break off, they dephosphorylate, right? So Fc gamma R2b can do that. And if I show you this diagrammatically, it might be obvious. Okay, so if the BCR there, which is a surface antibody, binds to its antigen, that causes cellular activation. If that antigen is in an IgG immune complex form, yeah, like I've shown there, i.e. it's got an IgG antibody bound, that will recruit Fc gamma R2b because this is an Fc receptor, it can bind to this bit of antibody. So what that does is actually recruit the phosphatases and switch off cellular activation. So you can see if you're a B cell and you've been activated, you've made lots of IgG, you're in the germinal centre, how does that ever end? Well, this is one way of switching off B cells, isn't it? Because once you've made lots of IgG, you'll have immune complex antigen, and that will switch this off. So this is a key break on B cells, okay? And it means, that without this, B cells can become out of control, and I'll show you the data to support that. So when I came to this as a youth then, as a young PhD student, full of enthusiasm as you are, this paper had just been published. So this was about the Fc gamma R2b knockout mice. So if you knock out this gene in a mouse and let them age, at about 6 months they get a nephritis that kills them. So they get autoimmune disease and they get a lupus nephritis. And they also found that if you used a whole lot of models, so for example, if you use a model of rheumatoid arthritis, which is called collagen-induced arthritis, these mice get a lot more severe disease. So what, you know, however you poke their B cells, without this receptor, they over-respond. So that's the knockout mouse. And then work that I started to get involved in, it turned out that in some of these spontaneous mouse models of autoimmunity, there was actually also a mutation in the Fc gamma R2b gene. So this was a mutation, a deletion, a 13 base pair deletion, sort of shown in that long yellow bit, within the promoter region of the gene. And I'm sure you know by now that if you have a genetic change in the promoter region of a gene, it may mean that that gene is over or under expressed, so it affects the expression level. So, and a 13 base pair deletion that overlay a site of transcription could be really important. And this was Nick Pritchard, who was in the lab when I came into the lab, found was that if you took various inbred strains of mouse that got autoimmunity, actually including the BXSB mouse, but also the NZBW mouse, and you stimulated their macrophages with immune complexes, that the ones that had this deletion in the promoter had a more active response to immune complexes. So this is just shown by calcium flux. So if you put on immune complexes to these cells, the cells get activated more exuberantly. And we also were able to show that B cells similarly were hyperactive following immune complex stimulation. And the critical thing was that the reason was, as we suspected, because this is a promoter polymorphism, if you look at the surface level of expression of Fc gamma R2b on mice with this deletion, it's lower, shown in red. So that the red thing, I'm sure you're used to looking at these plots. So that's a flow cytometry plot where you look at the surface level of expression of something. Okay, so it turned out when I went, one of the things I did was then went to look at a whole range of inbred strains. And in fact, that diagram that I showed you right at the beginning was a summary of my results. So it turned out that all sort of control inbred strains of mice that didn't get disease, didn't have the deletion and had high levels of Fc gamma R2b on macrophages and B cells. And the ones with lower levels had this promoter polymorphism and were autoimmune prone. And more recently, we've taken this further by saying, because you might say, well, therefore it's obvious that this deletion is what's causing disease. But to truly prove that, we used a genetic technique that's called a knock-in technique, where you make a knock-in mouse. And you can do this for specific human mutations too. So rather than saying we'll knock out the whole gene and what's the effect of that, we will actually knock in the part of the genome of your diseased animal or person that's different. And so that's the only bit that's different. And so what we did was the deletion, which you can see there in the promoter, we knocked that bit in, we've called that haplotype 1 in pink, but we joined it together so that the rest of the gene was a black 6 gene. Okay, so we're saying on a black 6 background that doesn't get autoimmunity, what's the effect of just putting that haplotype, that promoter polymorphism? And what we found using that technique was just having that tiny promoter deletion was sufficient for these mice to break tolerance and to be prone to autoimmunity. So here I'm showing you the anti-double-stranded DNA antibodies. And the knock-in mice, I should say, are in the white bars, and the wild-type mice are in the black bars. Okay, so we have evidence that Fc gamma R2b is involved in lupus from a knockout model, from a spontaneous model, and now a knock-in model. So what about in humans? And this is one of the things that that I became interested in. So a Japanese group had described that there was a SNP in the Fc gamma R2b gene. Now, unlike the SNP I showed you in the mouse model, which was in the promoter, this SNP, so it wasn't a large deletion, it was just a single nucleotide polymorphism within exon 5. And an exon 5 actually encodes for the transmembrane domain of the receptor. Okay, so that's quite an unappealing site for a polymorphism to be significant, okay, because the reason I say that if you have a polymorphism that affects the extracellular domain of the receptor, that might mean that it would affect how it could bind IgG. Yeah, so it might mean that it couldn't bind antibody, but it wasn't that. And if you have a polymorphism in the cytoplasmic domain of the receptor, it might mean that it affects the signaling capacity, so to make that receptor no longer able to recruit SHIP. But the polymorphism, this SNP wasn't in either of those, it was in the transmembrane domain, so that was a bit perplexing. But the reason these people had flagged it up was because in their study, they did a case control study. Yes, you take your patients with lupus, you take your controls and you say, what's the frequency of this SNP I'm interested in? And they found that, so the wild type form has got an isoleucine, right? But if you make this SNP, you actually get an amino acid substitution, so it's a non-synonymous SNP, and you get a threonine instead. So if you were homozygous for that, which I've termed as TT, so threonines, so you can see there was double the frequency of lupus, and that first publication was followed up by 2 subsequent publications looking at both Thai and Chinese lupus patients. So we wanted to answer 2 questions, so which were, you know, does this polymorphism actually affect the receptor function? Because that really tells you whether it's pathogenic or not. And if it does, how does it? Because like I said to you, it's a transmembrane domain polymorphism, and it wasn't obvious to us why it would impact on function. So to test this, we looked at a whole range of things. Now, this is a whole range of things that happen when you cross-link FC receptors on myeloid cells. Okay, so you get some signaling events, you get phagocytosis, so the antibody's internalized, and then that cell will produce inflammatory cytokines and co-stimulatory molecules. So I won't, the details don't matter, but what we did to test this was to take a human monocyte cell, so it was actually a cell line called U937, and we made that cell either express, so those cells at rest only have activating FC receptors. And we made that cell express either the threonine form of the receptor or the isoleucine form of the inhibitory receptor. And then we added immune complexes to the cell and said, you know, what does the effect of the threonine-containing inhibitory receptor have on activation? And this just looks at calcium flux. I'm showing you an example. So you can see wild-type cells that just have an activating receptor. When you put immune complexes on them at time 0, their intracellular calcium will go up, saying they've been activated. If you look at the B1s, which contain the native isoleucine-containing inhibitory receptor, that significantly inhibits the calcium flux. Yeah? It's an inhibitory receptor, that's what it should do. But with just a single amino acid change, so the threonine at position 232, which is what the B1T is, there is absolutely no inhibition of the immune complex induced response. So the response is the same as wild types. So by inference, you can say that these individuals at a cellular level are like Fc gamma R2b knockouts. You know, it's really, in this assay, not functioning at all. And we actually we looked at all of those things, and you can imagine that took a while, but found a similar thing, you know, more or less at a single cell level, both if you're looking at macrophages and B cells, they behave like Fc gamma R2b knockouts. So what's the mechanism of this? Because as I say, it doesn't affect antibody binding, doesn't affect signaling. So this got us to thinking about where signaling happens within membranes, and actually it occurs within these cholesterol-rich areas of the membrane called sphingolipid rafts, and that's where all of the signaling molecules, which I've shown here, so I know when I hear talks about immune signaling, it tends to make me sleepy, so I don't tend to talk about this in detail, but that's where all the signaling molecules are. And what we hypothesized was that if you had a threonine rather than isoleucine within the transmembrane domain, it would alter the electrochemical nature of the transmembrane domain such that it could no longer be recruited to the sphingolipid raft. So to test that hypothesis, we took some cells that were transfected with one or the other, and we isolated different fragments of the membrane. And so you can see that the sphingolipid raft bits, or the raft bits of the membrane, are in fractions 1, 2, 3 and 4. And if you blot for Fc gamma R2b, in the ones containing threonine, you can't find the receptor there within rafts. And similarly, we did some microscopy. So you can mark up rafts with cholera toxin B in red there. And you can see that the B1Ts don't co-localize. There's not so much orange as there is in the other. Okay, so to summarize that, these humans are like knockouts, and so perhaps it's unsurprising why they have susceptibility then to lupus. And this is looking at what effect it has on a single cell level. You know, I mentioned that the TLR7 people had gone off and looked at the effects in different cell types. So that's one of the things that I've done and I'm still doing. So I most recently published a paper on the effects of this mutation on dendritic cell function, on dendritic cell migration. And but I won't talk about that here. What I will say, if you stand back and think genetically, you know, why does this polymorphism persist? Okay, so it's clearly bad news. It predisposes you to lupus, but also to inducible autoimmune diseases of all sorts. So, if you were thinking about that, maybe it'll be helpful if I show you some other things. But could you, if you were thinking of a hypothesis, well, why do polymorphisms which clearly predispose you to autoimmunity still persist in the human race? So it can perhaps be helped if you look at the worldwide distribution of this thing. So one of the first things I did when I was a PhD student, so we'd looked at this promoter polymorphism in inbred strains. So we got some DNA from wild mice around the world. So there are these investigators, yes, truly. So there are these two French investigators that go and catch wild mice around the world. So which, I mean, I suppose it's not that bad, is it, going to, and they isolate DNA from them. So we were able to get some of these samples of DNA. And what you can see is that worldwide, what predominates is not the sort of what we thought was the wild type, but actually what predominates is the promoter haplotype that has the deletion in it. So the autoimmune prone promoter predominates. And in fact, what you can see, going back to my Japanese mouse fanciers story, was that, you know, is actually that wild type non-deletion is present in wild mice there, and that's why it's come as being what's present in our inbred strains, because that's where they come from. But if you look worldwide, it's actually the autoimmune prone promoter that is dominant. And if I show you, so this is now the human polymorphism, and I'm showing the frequency of it in different populations. So in Caucasians, we know that the TT polymorphism, or the TT genotype, is relatively rare. It's actually about 1%, 1 to 2% of Caucasian populations. But if you look in other populations, so for example some African populations, you can see 11% of individuals are homozygous. And similarly some other Southeast Asian populations. So we hypothesized that the reasons these polymorphisms had persisted, and actually had persisted at a high level in some populations, was that it protected against infection. So they'd actually been selected for that reason. And so I went on to test this in 2 models. And we chose these 2 models because both of them antibodies play a role, and both of them are big killers worldwide, and what they kill are infants. So that is, they kill you before you can pass on your genes. So if you have something that protects you against these, it's likely to have a very strong evolutionary impact. So strep pneumoniae is the commonest cause of community-acquired pneumonia in the UK, but worldwide kills millions of infants. It can also cause a very severe meningitis in children and peritonitis. So what I did was to take the wild type, an Fc gamma R2b knockout mouse, and I gave them some strep pneumoniae. And what we found, I could titrate this, so we've got the mice on 2 different backgrounds, such that the strep pneumoniae would kill all of the controls, but 20% of the knockouts would live. So you can see in the evolution, that means you're selected or not. So that would be a big thing. We could show other parameters that it doesn't matter. So what about in humans? Well, what we were able to do, one of the cool things we have here in Cambridge is called the Cambridge Bioresource, where we're able to recall healthy individuals based on their genotype. So they've all come to give blood and have agreed to be part of this. So the different investigator goes and says, well, I think I have a polymorphism that's important. I'd like to get some cells and look at them. You can recall them and then do the test. So when we started to optimize these tests, we used our own blood. So I know that I'm a very dull II genotype. But we did have someone in the lab who was a het, who was very popular. But this resource has meant that we don't have to cause anemia in postdocs anymore. But what you can see, what I did was to take monocytes. I gave them some MCSF, which makes them into macrophages, and I gave them some antibody opsonized strep pneumoniae. And the TT homozygotes are able to take up much more of it. And they also produce more TNF-alpha and IL-6, which help to protect you from it. So we then looked at malaria, so really up there is a very strong evolutionary force, and I showed you the distribution of the genotypes is strongly suggestive that it might be important. So again, we did the mouse model, so I actually went off to MPhil to do this in Jean Langhorne's lab. So again, wild-type and Fc gamma R2b knockouts, and you infect them. And then you do, so I would say that much of basic research, which is, might be ahead of you, is quite tedious, and this is particularly tedious. So what I did was to take these mice at day 6 and day 8 shown here, nick their tails, get a little bit of blood, put it onto a film, and then you sit in a room, and in one hand I had a counter where it was total red cells, and the other hand a counter that was parasitized red cells. So you can see the little parasites, ring-form parasites here. So this gave me repetitive strain injury, and I told my supervisor I'd be suing him, but I didn't in the end. But so much of research can be dull, but what we were able to show was that the Fc gamma R2b knockout mice had lower parasitemia levels, and we could similarly do phagocytosis assays showing that their macrophages were better at taking up opsonized parasites, and that they could produce more TNF. And we then went on to again get monocytes from individuals with different genotypes. And here this was a tricky experiment because we used falciparum here. You can't use a mouse strain of malaria for human cells. They're very specific to species. So Lisa Wilcox, who's in the lab, went over to Oxford to collaborate with someone called Britta Urban, who grew up falciparum, which is actually very dangerous if you get an infection. And then we fed to these monocyte-derived macrophages antibody-opsonized parasitized red cells. So, and what you can see is that what we used to opsonize the parasitized red cells was either non-immune serum from EU subjects, okay, or immune serum from children in Kilifi in Kenya, where one of the Wellcome Trust research centres is, or immune serum from adults from the same place. And the individuals, the TT homozygotes, you can see this is looking at phagocytosis, that they have the highest phagocytosis, and that's particularly true if we look perhaps here, although is not doesn't look necessarily majorly significant. One of the problems with using humans is that the error bars are always very wide. But you can see, interesting, we also found that possibly heterozygotes had an advantage too, which again may explain some of the selection. And then to nail this, Lisa then went on to do a genetic study where she looked at genetic samples that had been obtained from children in Kilifi in Kenya that had gone in with severe malaria. And if you compare the frequency of TT homozygotes in those with severe malaria and those with controls, so if you just look at because we did this in a replication cohort as well, you can see in severe malaria cases, the TT homozygotes are underrepresented, about half of them in there. So it strongly suggests that this is protective against severe malaria. So when we think about the impact of any genetic polymorphism that affects the immune system, yes, what's driven it, although now, in our very clean environment, the phenotype that we might see is autoimmunity, what's driven its selection is actually this interaction with pathogens. And this was actually the first immune gene that had been identified that a naturally occurring polymorphism has been selected in humans in that way. Okay, so we're on the final straight, so I hope you forgive me for that mild bit of digression, but I think it shows you a sort of bigger picture of why some genetic polymorphisms are selected. Okay, so we're on to the final straight. So one of the things that's been focused on in understanding the pathogenesis of lupus is why B cells are abnormal, because clearly they're at the centre of this because they are the precursors of antibody-producing plasma cells. So you can see that there could be problems at a number of levels, and in fact it turns out that in patients with lupus they usually have polymorphisms that result in a little bit of all of these. So they must have autoreactive B cells, they must have autoreactive T cells, and in fact it turns out that they probably have an excess of T cell help and also a failure to control activation of B cells, and I showed you very nicely one example of that in Fc gamma R2b. But what's the evidence that B cells are truly central in lupus? Well, you know, why can't it be that you just have T cells that are excessively driving this? So again, some of these mouse models have been used, and particularly I'll show you some data with the MRL mouse model. So people made a version of the MRL mouse that was unable to secrete antibody, but still had B cells. And they were able to show that in those mice, unsurprisingly, so this is the JHD MRLs, they had low levels of antibody in their serum. So then you can say, well, what's the effect here? Is the effect that the B cells are having, is it just about making antibody, or is it something else? So when they looked at these mice, you can see that they actually have lower levels. So this is the score of lupus nephritis, lower levels of all of those. So they then re-expressed antibody in these mice and showed that in those mice you could actually restore disease. So this is looking at different versions of mice with no B cells, no antibody, B cells and no antibody, B cells and no antibody. And the conclusion of these experiments, making mice with either that were on an autoimmune prone background but either had no B cells or no B cells, no antibody, was that B cells contribute, both is not just about antibody production, but they also contribute more generally to the breakdown of autoimmunity. And one of the key reasons for that is that they can be potent antigen presenting cells to T cells. And I say that because I'll come back to it, therapeutically, one of the things we do know we can do is to deplete B cells. And we have monoclonal antibodies that can do that. Okay, so evidence for abnormal T cell help. Well, the best, or I would say that one of the most convincing mouse models of this, what's called the San-Roch mice. So these were, and if I had time, I'd tell you, I would have put a few more slides in about this. But so another way that you can identify genetic mutations that cause disease in mice is that rather than, you know, in the inbred strains, basically you're relying on some mouse fanciers breeding them a lot and it happening. What you can do is introduce a mutant, a genetic mutant, as in an agent that, or what I mean is a mutagen. You can introduce something into the mouse's diet that will increase the likelihood of mutations being introduced. And people have done that, so using ethyl nitrosylurea, which if you put in the drinking water of mice, it makes them get more mutations. And then if you screen those mice, and this is one of the mice that came out of that screen, and these San-Roch mice, so they were named this after Saint Rocin, who apparently, or Santa Roque, who was apparently the patron saint of the bubonic plague. So these mice had massive buboes, so they basically had huge lymphadenopathy. So in the whatever gene was being knocked out in these, or being mutated in these, was causing B cells to excessively proliferate and to have massive lymph nodes. And it turned out that the pathway that was being affected was the ICOS pathway, and that ICOS pathway we now know from mouse models, and I put the references here if you want to look it up, are critical for the survival and development of T follicular helper cells. So if you drive the formation of these T follicular helper cells, you can basically, even if you've got a normal B cell, it's sufficient to drive lupus. And so when they went on to look at the kidneys of these mice, so you can see the Rocin mice on the bottom have got lots of, the purple dots are basically immune cells infiltrating the kidney, and they were also able to score that, so you get a pathologist to look at it and say how much nephritis is there, and you can see that the San-Roch mice have got high nephritis scores, and even the heterozygotes have got less nephritis. Okay, so that's all of those things we've done so far. Autoreactive B cells, abnormal T cells. What about controlling the activation state? So one of the key inputs, I said, you know, if a B cell sees an antigen via its BCR, it doesn't necessarily get activated. It relies on having extra signals, so for example, complement. And one of the extra signals comes via a cytokine called BAF, or B cell activating factor of the TNF family. And it essentially, there are 3 receptors for it expressed on B cells at different time in their development, but it drives the survival of naive B cells and augments their activation following stimulation. So it's been known that in patients with lupus, if you sample their blood, you can actually find high levels of this cytokine BAF in some individuals. And so to look into this more carefully, a group made a mouse that just overexpressed BAF tonically, so it wasn't appropriately expressed. You introduce a transgene, which means that this mouse has very high levels of BAF, so maybe recapitulating what you can see in humans with lupus. And what they found was if you looked at the spleens of those BAF transgenic mice, they had higher spleen weight, and this was because they were packed with even more B cells. And if you looked at their kidneys, so this is just proteinuria as a surrogate of nephritis, you can see that they had nephritis. But what was one of the key things about this follow-up paper here in 2007 was that they made a version of the BAF transgenic mouse that lacked T cells. And what they found was that in the mouse that BAF transgenic mouse that lacked B cells, so this is the fourth bar, yeah, the disease you got was the same as in mice that had T cells. So what it tells you is if you drive the co-activation of B cells hard enough, B cells begin to function autonomously and no longer need T cell help. So this is really critical. So you can see, again, if we go back to the polymorphism idea, if you have an individual who has a polymorphism in their BAF gene, which means they produce higher levels than average, it's just another brick in the wall that means that they might go on to develop autoimmunity. Okay, so how have we applied this finally? Well, there's good rationale to target B cells based on what I've told you. And I think the other thing that's encouraged us to go down this route is that we know that if we deplete B cells, what we might do is make someone immunodeficient in that they would not be able to produce antibody. But we know that there are individuals who have genetic abnormalities such that they never produce antibody, and we can help them. We can actually give antibody back to them. So that's not something that we can't overcome. So there are a number of monoclonal antibodies that have been used. So, and I would say the one that's probably been most used is something called rituximab that recognises a protein that's found on the surface of all B cells, the CD20. And rituximab is extremely effective at depleting B cells. We have some other antibodies. There's one called erythematosus that recognises CD22. And then there's some in development that recognise CD19, but the whole idea being, can you deplete B cells? And if you, so it's just a bit about rituximab, which I've told you about, but if you do, so this is a summary of all the studies that have used B cell depletion in lupus nephritis. And the summary is that it's extremely effective, more effective than the things we've had previously at treating lupus nephritis. So complete response is shown in the green blocks. There was a randomized controlled trial to use rituximab which didn't show statistical significance, so a phase 3 trial, which is a bit sad for the people that had organised it. But I think there was a lot of criticisms of the inclusion criteria, so for example, it turned out that quite a large proportion of patients they'd included were ANA negative. So what sort of lupus did they actually have? So I think although the phase 3, the gold standard evidence for using rituximab in lupus isn't great because of that trial, most clinicians do use it. But what has been more effective in targeting clinically is the BAF pathway. And one particular drug, so an antagonistic BAF antibody called belimumab that's made by GSK, has been used in a number of trials, and I gave you one of the key phase 3 trials in your reference list. And basically, the data using belimumab shows that it's certainly more effective than standard of care, which is the things I mentioned like cyclophosphamide. And obviously comes without the price for the future of an impact on future malignancy. And I should have said also cyclophosphamide has severe effect on fertility. And since I told you that 9-to-1 female to male ratio, that makes a big difference. So this has really been big news. So belimumab is the first new drug to be licensed for lupus in 50 years. So this was a real turning point in the treatment of lupus. Okay, so we've made it and there hasn't been too much sleepiness. So hopefully I've convinced you that lupus is clinically heterogeneous, and it's such an interesting disease that I think there possibly might be more people investigating its pathogenesis than there are people who have it. Both genetic and environmental factors contribute to disease susceptibility, and I could have given you a whole lecture about the environmental factors, perhaps another time. We've been able to use spontaneous mouse models and mouse knockout models to identify genes and polymorphisms. And I would say more than any other disease, they've been just so useful in identifying underlying pathways. What we do know is that B cells play an absolutely central role, and this has actually made a difference to patients because we've been able to begin to target B cells with monoclonal antibodies to produce treatments that are more specific and, more importantly, less toxic for the young women that get the disease. So there we go.